Okay, so welcome back to this next video in which we are discussing the phospholipase C beta family of enzymes. Okay, so we've now discussed the structure of a phospholipase C beta enzyme. We've discussed how the inactive enzyme is continually moving between being in the cytoplasm and being attached to the underside of the phospholipid bilayer. What we now want to discuss is how those phospholipase C enzymes which are attached to the underside of the phospholipid bilayer, when you activate a G protein cascade, are going to get activated, basically. We want to discuss how these are going to be activated by the alpha subunits of GQ/11 heterotrimeric G proteins. Okay, so we're going to start off with a discussion of G protein coupled receptors. We'll then move on to heterotrimeric G proteins, then we'll discuss the G protein cycle, and then we'll discuss actually how an alpha Q/11 heterotrimeric G proteins alpha subunit is going to activate these phospholipase C beta enzymes. Okay, so we're going to start then with GPCRs, G protein coupled receptors. Okay, right. So let's start with the different portions then of a G protein coupled receptor, because this will be important in our understanding of how they're going to interact with heterotrimeric G proteins. So, G protein coupled receptors are a massive, great family of proteins. There's around 800 G protein coupled receptors just in humans alone. Okay, and those are different types of G protein coupled receptors. Okay, so the conserved feature that they all have is that they have 7 membrane-spanning alpha helices. Now, even though I'm drawing these membrane-spanning alpha helices just as straight lines, in reality they would be alpha helical in structure. Okay, and they have their amino terminus extracellularly and their carboxylic acid terminus intracellularly. Okay, so let's just discuss the nomenclature for the different domains of a G protein coupled receptor. So we'll start off with the 7 membrane-spanning alpha helices, which I'm now highlighting in purple here. Okay, so the first membrane-spanning alpha helix here is known as TM1 for short, and that stands for transmembrane alpha helix 1. Okay, then the second one will be called TM2, the third one TM3, the fourth membrane-spanning alpha helix TM4, the fifth TM5, the sixth TM6, and the seventh will then finally be TM7, and I'll just label up the first and the last, because if I try and label up all of them it will get tedious and also very crowded. Okay, so let's now talk about other domains other than the membrane-spanning alpha helices. You also have, and very important for the interaction with heterotrimeric G proteins, you have these intracellular loops between the membrane-spanning alpha helices, now highlighted in orange. So we have 3 of these, one between the first and the second membrane-spanning alpha helix here, one between the third and the fourth membrane-spanning alpha helice here, and one between the fifth and the sixth membrane-spanning alpha helices. Okay, so these collectively are known as the intracellular loops. Okay, and for short, the intracellular loops are often abbreviated to ICLs. Okay, so I for intra, C for cellular, L for loops. Okay, right, so this first intracellular loop between TM1 and TM2 will be called ICL1. This second intracellular loop between TM3 and TM4 will be called ICL2, and this third intracellular loop between TM5 and TM6 will be called ICL3. So we have ICL1 to 3, basically. There's similar nomenclature for the extracellular loops that I'm now highlighting in blue here. Okay, so here we have 3 extracellular loops, and these are abbreviated in short to ECL, so E for extra, C for cellular, L for loop. And you have the first extracellular loop, which is between TM2 and TM3, then the second extracellular loop between TM4 and TM5, and then finally the third extracellular loop between TM6 and TM7. Okay, so those are the extracellular loops. Now the final 2 domains to discuss are firstly this domain that's prior to the first membrane-spanning alpha helice and is extracellularly. Okay, that domain is known as the amino terminal domain. Okay, and for short, the amino terminal domain is often abbreviated to the ATD. Okay, in addition, some people will actually call the amino terminal domain the amino terminal ecto domain. Okay, so some people will put an ecto in front of domain there, because this domain is on the outside of the cell, so that's why it's referring to ecto domain. Ecto means on the outside of. Okay, and for short, they'll abbreviate amino terminal ecto domain to ATED, like so. So A for amino, T for terminal, E for ecto now, and D for domain. Okay, so you might see that used. So those are all referring to this region in red then, here. Okay, and then finally you've got the domain that is after TM7 down here, okay, and that's known as the C-terminal domain. And for short, the C-terminal domain, as with the phospholipase C beta enzymes, is abbreviated to CTD. So this is the C-terminal domain. Okay, right. So that's the different bits of a G protein coupled receptor then. We now want to discuss heterotrimeric G proteins, because when the ligand comes and binds to the G protein coupled receptor, what's going to happen is the G protein coupled receptor is going to change conformation. Specifically, the 3 intracellular loops here are going to change conformation so that they can now bind to the alpha subunits of heterotrimeric G proteins. So let's now discuss heterotrimeric G proteins then. So the name gives us some clues to the structure of heterotrimeric G proteins. Okay, heterotrimeric means a trimeric structure is a structure that has 3 subunits making up the entire thing. Hetero means different, so this tells us that the 3 subunits that make up the full complex are going to be different subunits. Okay, so let's draw a heterotrimeric G protein then. So we'll begin with the most important of the 3 subunits that make up a heterotrimeric G protein, which is the alpha subunit. Okay, so these 2 lines here represent the phospholipid bilayer once again. Okay, so here is my alpha subunit here. Okay, so the alpha subunit of heterotrimeric G proteins is very special because it can be in 2 states. It can be in an on state, in which it has guanosine triphosphate GTP bound to it, or it can be in an off state, where it has guanosine diphosphate GDP bound to it. Okay, let's say initially that our alpha subunit is in the off state and has guanosine diphosphate GDP bound to it. In addition, let's colour in our alpha subunit in red here. Okay, right. So at the moment our alpha subunit is in the off state. Now, the other thing I need to discuss is the fact that the alpha subunit is attached to the inner leaflet of the phospholipid bilayer. It is not an integral membrane protein. It's not sitting within the membrane, but it is going to be attached to the inner leaflet of the phospholipid bilayer. Now, how does it achieve this? Well, basically, alpha subunits have lipid moieties attached to them. Okay, so they have a lipid group attached onto one of their amino acids, and this lipid group then sticks into the inner leaflet of the phospholipid bilayer and holds the alpha subunit there. So basically the idea is this, that the alpha subunit is a single polypeptide. What you're then going to do is attach onto this via one of its amino acids a long chain carboxylic acid, which will stick off the side. Okay, and this will be extremely hydrophobic, so it will end up implanting into the inner leaflet of the phospholipid bilayer, and hence it will hold the alpha subunit dangling below the inner leaflet of the phospholipid bilayer. Okay, so what examples of lipid moieties can you attach to alpha subunits of heterotrimeric G proteins? Well, every alpha subunit gets palmitoylated. Okay, so let me show you what palmitic acid is. Okay, right. So palmitic acid is the old name for a molecule that would now be called hexadecanoic acid. And it's another example of a long-chain carboxylic acid. It's got a very similar structure, in fact, to stearic acid, octadecanoic acid. Okay, this name tells us that we're dealing with a fully saturated 16-carbon carboxylic acid. Okay, so here is our carboxylic acid group. Then we need another 15 carbons, so we need 14 methylene groups then. So here's one methylene group. I'll bracket it and put 14 there. And then on the end we have a methyl group. Okay, so this is the structure of palmitic acid. What you can do is you can attach palmitic acid onto residues within the polypeptide of the alpha subunit, and these palmitic acid molecules will stick off the side of the alpha subunit, and they'll have an extremely hydrophobic tail, and that hydrophobic tail will implant into the inner leaflet of the phospholipid bi there. Okay, and we'll call it there for this video, and we'll continue this discussion in the next video.